Welcome to video 8 of Genetics the Code Broken. In this video, we're going to be looking at gene therapy, how it can be used, and we're going to look at one of the examples before you undertake your own secondary source research task into finding another use of gene therapy. So gene therapy is the treatment of diseases by replacing, switching off or correcting faulty genes that cause diseases. So it takes 4 steps. So first, the gene that causes the disease needs to be identified, and then they need to locate the cells or tissues involved in the disease. There needs to be access to multiple copies of the normal gene, and then this normal gene needs to be inserted by a delivery technique to the affected cells. So the first few steps, so actually identifying the gene and location of these cells and tissues, is actually, and the access to the multiple copies of the normal gene is actually the simple part of the process. The part which is taking the most to kind of perfect is in fact this insertion, the techniques which we can use to insert these genes. So they've tried these 5 processes. So the first is using viruses as vectors to carry the replacement gene into a cell. They've tried direct injection, where they've injected DNA containing the normal gene into cells, linking the normal gene to a molecule that will bind to a receptor on the surface of a cell and then pass it into a cell. Nanotechnology has been used to create an artificial lipid sphere, which contains the normal gene that can pass through the cell membrane. And the last is by constructing an artificial chromosome carrying the normal gene for insertion into the cell. So what have been some of the problems with some of these techniques so far? The incorporation of the inserted gene into the cell's DNA does not happen easily, and there is no control, in fact, over where the new DNA would join the existing genetic material. The use of viral vectors has appeared to be the simplest process, but it causes issues as the viral vectors themselves are pathogens, which can, 1, cause harm to the patients, or 2, the patient's immune system actually will destroy the virus, meaning that this gene that has tried to be inserted actually doesn't get used because it's been destroyed. The cells in which are being targeted may have a limited lifespan, which means that they might incorporate the working part of the gene, but then when they actually die off, it means that the treatment needs to be repeated very often to maintain that health. And then there are ethical issues with the use of this technology and this process. So one of the examples is haemophilia, and haemophilia is a sex-linked inherited blood disease, and there are 2 types. So in type A, it's missing factor 8, and in type B, it's missing factor 4. And these factors are essential clotting factors, which causes a person to bleed easily and for longer than normal. So the traditional treatment has been injections of the missing factor, so that this factor is just injected into the person, and they'll have to have that throughout their life. But in terms of starting to use gene therapy, they've tried transferring just the factor 4 gene by vectors. Initial trials saw some temporary improvement in the clotting, but caused side effects. And in current trials, they're using the nanoparticles which contain the gene, and they're being injected into the liver cells to be used by the body. This is where also we start to look at stem cells and how they can be used to aid gene therapy. And so stem cells are cells that can develop into different kinds of a tissue in an organism. They're cells that haven't differentiated yet, and they have a great potential for use in curing diseases and, in fact, for cloning purposes. So if we can manipulate them in vitro before being retransplanted into a patient by injection into the bloodstream, and then once they are injected, they're able to travel to the place in the bone marrow where they are functionally active, and then these cells are able to contribute to the mature blood cells and be used by the recipient for an extended period of time. So here we can see just how these stem cells might be used. So we can see that the gene can be inserted as a retrovirus and then introduced into these stem cells, which are then reintroduced into the patient. Or we can see the direct delivery, which is injecting that virus with the therapeutic gene straight into that liver. So we have the direct delivery and the cell-based delivery here. That concludes video 8. Make sure you tune in for video 9.